Welcome to the next video in the Search for Better Health topic. This video will be looking at two syllabus dot points. 9.4.43, explain why organ transplants should trigger an immune response, and 9.4.54, outline the reasons for the suppression of the immune response in organ transplant patients. So we can see from the two syllabus dot points that they actually come from different focus areas or different sections of the syllabus. However, because they're both related to organ transplants, they fit together quite well. So the body has an incredible ability to distinguish self from non-self. So we've been looking at the body's ability to carry out the immune response. And the T lymphocytes of the immune system can recognize the body's own cells so that they are not destroyed and can recognize foreign cells and destroy them. This recognition is due to the ability to bind or join to the foreign cells. All cells have chemicals on the surface of their membranes that are used for this recognition. They are mainly glycoproteins, and they are called cell surface molecules, and lymphocytes do not react against them, so they do not bind to them or destroy them. There is one group of molecules, however, that can pick up antigens and can trigger the activity of T and B lymphocytes, and they are our major histocompatibility molecules, or our MHCs. So we've talked about them a little bit already in looking at the immune response. So these guys are rather complicated. So their name comes from the words histo, which means tissue, and compatibility, which means to be able to live in harmony together. So basically, information for making these molecules is on a cluster of genes called the histocompatibility complexes, hence the term MHC. So basically, these guys work in order to make sure that all of the tissues in our body are working together properly. So MHC1 molecules play a major role in recognizing the body's own infected cells, while MHC2 molecules are more active in recognizing the foreign cells or the pathogens themselves. In both cases, the antigen from the foreign cell is recognized either on the surface of the body's own cell or on the surface of the pathogen. The result is that the infected cells or pathogens are destroyed. In this role, the MHCs act like holders, a bit like a candlestick holding a candle for the antigens from other species, including bacteria, viruses, and fungi. Now, a slightly different situation arises when a non-self cell from another individual from the same species enters the body, such as an organ transplant. So the new organ that they are receiving from another person has marker molecules or antigens on its surface that are different to the ones that we produce ourselves. So what happens is the transplanted organ is identified as foreign, and the immune response is activated in order to defend the body. So that ties directly back to our syllabus dot point, explain why organ transplants should trigger an immune response. So that's exactly what this is here. So the body identifies the marker molecules as being different, therefore antigens, which then leads to our immune response. So tissue transplants are carried out between individuals who have very closely matched tissue MHCs. However, there are hundreds of different possible alleles for the genes controlling MHC1 and MHC2, so it is very unlikely that two individuals chosen at random will have similar MHCs. So in fact, a person's MHC pattern is a bit like their tissue fingerprint, so each person's is very different. So that term allele will come across when we do the blueprint of life topic, and that basically is the different versions of the genes. So there's a number of different versions of the genes that control both MHC1 and MHC2. So even after careful searching, the match between the tissue of the donor and the recipient, if you've ever experienced or heard of people who are undergoing an organ transplant, there's quite a considerable amount of time that is spent looking for a suitable donor. So even after that lengthy process, the MHC molecules may not be exactly identical. So what happens is the recipient's immune system must be suppressed in order to minimize rejection, which goes back to our second dot point that we're looking at. So as we've mentioned, marker molecules ultimately initiate the immune response, and that's why we need to suppress the immune system in transplant patients. And what happens is once the immune response is initiated or it's started, the cytotoxic T cells are activated and move to the transplanted organ to attack and destroy the cells. So just like any other foreign cell or any other pathogen that's entered the body, the cytotoxic T cells identify those cells as being different, so it actually starts to kill the cells in the transplanted organ. So this causes rejection of the transplanted organ, so the body starts to fight against it and then basically breaks it down and tries to get rid of it. The big thing to note, however, this only happens with the cytotoxic T cells. Our macrophages and B cells do not attack transplanted cells. So, excuse me, the immune system is suppressed by the administration of immunosuppressive drugs. So obviously the term immunosuppressive, so stopping or suppressing the immune system. So modern drugs such as cyclosporin can suppress part of the immune system that causes the rejection while leaving other parts of the immune system active. So these drugs act to reduce the activity of the killer T cells, as these are the principal cells that attack the transplanted organs. This is the advantage that the whole of the immune system is not suppressed and can still act to defend against other pathogens. The immune response will not be as effective, obviously, in people who are taking immunosuppressive drugs because the interactions between the B and the T cells cannot occur. So just like we looked at with the immune response, the B and the T cells need to sort of trigger each other to carry out the whole cycle of the immune response. This means that the patient is at greater risk of suffering from other infections. These anti-rejection drugs must be taken for the rest of the patient's life in order to stop the immune system from detecting the transplanted organ and destroying it. And they are sometimes needed in large doses at first, but over time the immune response lessens and therefore lower doses are needed. And that brings us to the end of this video. So we've looked at why transplanted organs firstly create an immune response, and secondly, how immunosuppressant drugs help to stop that rejection of the transplanted organ. And that brings us to the end of this video. Thank you for watching.